foreign pathogens and parasites by these two immunity mechanisms, activating these two types of mechanisms. And these two types of innate and adaptive immunity branches can also help fight against or fight over different types of pathogens like bacteria, viruses, fungi, and parasites like protozoan parasites and helminth parasites. So, uh, to, uh, first, coming to the innate branch of uh, immunity, you know about innate. What is exactly innate branch or innate immunity? It comes by birth or inherited by our parents. So, the barriers, especially different types of barriers, prevent the entry of the different types of pathogens and parasites in this branch of innate immunity. So, we have physical barriers, okay, and chemical barriers and physiological barriers and different types of cellular barriers. Okay, so coming to the bacterial uh, immunity of infectious diseases against bacteria. So a major feature of innate immunity is the presence of the normal gut flora, which can competitively inhibit the binding of pathogens to gut epithelial cells. So these gut epithelial cells uh, have different types of receptors and also having the mucosal layer on the surface of this epithelial cells. So this mucosal layer contains different types of uh, the physiological barriers, like it contains acids, okay, and pH also vary from other areas to this mucus layer. So innate response also can block the establishment of infection. It blocks the establishment of the infection. It cannot foreign pathogens cannot able to attach with the or enter through the epithelium by these layers. Okay, these the barriers. So it prevents the infection. And for example, gram-positive bacteria contain a peptidoglycan that activates the alternate complement pathway. So this, if the foreign particle or pathogens get entered into the epithelial layer, different types of mechanisms, okay, immune mechanisms also protect or prevent the um, proliferation, okay, growth of the bacteria by activating the different types of other branch of immunity mechanisms. So, and all the surface molecules of foreign pathogens and parasites are released or products of the foreign pathogens also act as the antigens. Those surface molecules and products of pathogens are acts as the antigens and uh, recognized by our immune cells and other molecules. So by these pathways and by this way, different types of endogenous and exogenous pathogens are uh, recognized. And coming to the one by one, we will discuss one by one. First, I would like to talk about 1 minute virus. Okay, I would like to talk about virus firstly. So, as per your book, you have virus immune response. Okay, immune response against virus. Okay, I will change this image. Just wait for me. Wait for me. I'm going to save one image. So, This viruses infect our cells and by the contacting with the cell, and also this virus having the different types of RNA molecule, genetic material, also transported into our cell. So by these different ways, error will. So the different types of viruses uh, adhere or attach to the host cell, and after attachment with the host cell with the different types of receptors, especially virus contains glycoprotein molecules on the surface of the layer of the virus. Okay, and these glycoprotein molecules are especially 120 glycoprotein GP120, GP41. With the help of these molecules, uh, virus adhere or attach to the on the surface of the host cell, especially. So attachment in the attachment, these proteins, glycoproteins will help to attach to the target cell, and after the attachment, viral viral genome can be inserted, will be inserted into the uh, host cell or target cell, and with the help of different types of enzymes. Okay. Viral genome and enzymes are released following removal of the core proteins. Okay, after the releasing of the genetic material, remaining portion will be released. Uh, the remaining material, rather other than genome material of virus, will be released after inserting of the genetic material into the target cell. So viral, after entering into the uh, host cell, viral genetic material inserted will be inserted into the normal DNA. Okay, by the after conversion of the uh, double stranded, okay, proviral DNA. Okay, after conversion of the RNA into the DNA by the process of reverse transcription. Okay, the conversion of RNA into the DNA will be occur. Will also be happen with the help of different types of enzymes. Okay, this process is known as reverse transcriptase, reverse transcription. Okay, enzyme is reverse transcriptase. Okay, in this process, RNA, viral RNA, converted into the DNA molecule, and after the conversion of the uh, DNA from RNA, and this DNA will be inserted into the DNA. That is known as integration. Okay, integration. Okay, integration of the viral DNA molecule will be happen after the transcription. Okay, reverse transcription, and after the integration, along with our or along with the host cell DNA molecule transcription, viral proteins, okay, viral molecules also, viral genome also be transcripted along with the host cell DNA transcription. Viral mRNA also be produced after mRNA production. The translation of the different types of protein molecules are happening, and after the translation, the assembly of different proteins may produce the new viron, okay, or new virion. Okay, and this is a simple life cycle of the virus after getting into the infection of the cell. And you know about this different type of viral life cycles. So what happens after infection to the cell? What happens to the host cell? Okay, what is the host cell reaction? Okay, response to the cell or host body response against the infected viral infected cell. That is the important aspect for our immunology topic, today's topic. The first step uh, into the you know about the different types of virus mechanisms, different types of immune responses like innate immunity, and we will discuss everything today, innate immunity and humoral immunity, cell-mediated immunity, everything will come under different types of responses, immune responses. So, what happens? How our cell respond? How our body immune system respond against the viral particle? Whenever the viral particle comes into and contact with the uh, our body or cell, what is the immune response against our uh, against viral particle? That is important. Okay, and whenever this body get our cell get encountered with the uh, cell, and our body fight against the different our body system fight against the different viral by the different immune mechanisms. For example, humoral immunity. Okay? One of the important and effective response of our immune system is that humoral response with antibody, especially secretion of immunoglobulin A. Okay, secretion of immunoglobulin A from the B cells. Okay, and these are the antibodies. We can also call as secretory antibodies. Play a key role in the blocking of the binding of virus to host cell and thus preventing the infection or reinfection. Okay? And whenever our cell get infected with the viral material, and this all the viral material presented by the all infected cell presents these antigens with the help of MSC1 molecule, and this MSC1 molecule present the antigen to the TC cell, TC cell, and this TC cell directly kill the cell. Okay, that is the one of the mechanism. And sometimes this MSC antigens also uh, bind with the help of B cell, and B cell produces this type of the immunoglobulin A molecules. And after binding with the antigen with the B cell, will stimulate the proliferation of the B cell and the production of the immunoglobulins will be stimulated after the proliferation of the cell. And these immunoglobulin A molecule goes and binds with the uh, host cells, okay, virus host cells, thus prevent the infection of the infection and reinfection. Okay, binding virus host cells, okay, virus host cells with the help of these antibodies, that reinfection will be inhibited. And some other antibodies also there, immunoglobulin G, immunoglobulin M, and immunoglobulin A antibodies are released uh, against this type of viral antigen. What happens after releasing of these immunoglobulin G, M, A from the plasma cell? And these antibodies go and block the fusion of viral envelope. Okay, and the first step in the virus life cycle is that fusion, okay, adherence or attachment to the host cell. That can be inhibited. The first step of the viral life cycle can be inhibited or will be inhibited after fusion of these immunoglobulin GMA to the host cell. And this fusion of the viral molecule or viral protein layer with the plasma membrane can be inhibited after binding of the immunoglobulin GMA. This is one of the important steps in the humoral branch of immunity. And coming to the other specific features in the humoral branch, immunoglobulin G and immunoglobulin M antibodies also enhances the phagocytosis of viral particles. Okay, these immunoglobulin G and immunoglobulin M molecules are having the receptors for the complement proteins, and whenever these antibodies goes and binds with the antigens of viral particle, and this antigen antibody complex attracts the different types of complement proteins. Okay, and after attachment to the antibodies receptor with the help of complement protein, it enhances the opsonization process, or it helps to the activation of opsonization. Opsonization may lead to the enhancement of the phagocytosis of the infected cells. Okay. Overall, immunoglobulin G and M play a role in the elimination of or killing of the infected cell. Okay, especially viral infected cells. So, immunoglobulin M antibody particles. Okay, agglutination of different types of viral molecule or viral antigens also taken place with the help of immunoglobulin M. And complement activation. Okay, complement, I have told you earlier, complement activation and complement, whenever this complement activation is taking place with the help of immunoglobulin G and immunoglobulin M antibodies, and especially with the help of C3b, okay, subunit of C3 complement protein. And this C3b is the mediator of opsonization of different types of phagocytic pathways. Okay, whenever this CCB subunit go and bind with the immunoglobulin G molecule and immunoglobulin M molecule, which are having the receptors for the C3b on its Fc region, and this binding of the complement protein, especially C3b, to the antibody G and M, will attract the macrophages, okay, which are having the Fc receptors on its surface. Example, dendritic cells and macrophages are having the immunoglobulin G, M, or Fc receptors on its surfaces. Okay, complement binding with the Fc receptor of the G and M antibodies attracts the macrophages towards the infected sites, and engulf of the different types of infected cells will be happened after the opsonization process. And this immunoglobulin, this is comes under humoral branch of immunity. So this all these immunoglobulin A and M, G play a key role uh, to fight against the uh, virus particles. Okay, this is brief account of immune response to viruses. And coming to the cell-mediated immunity, okay, cell-mediated immunity branch in that immunity branch, and different types of cells releases. Okay, different types means the in this branch different types of cells are there. For example, TC cell, TH cell, B cell, and natural killer cells. Different types of cells are there. So the interferon gamma secreted by T helper cell or T cytotoxic cell has direct antiviral activity. Okay, whenever the antigens presented by the dendritic cell or macrophages, and these dendritic cell macrophages are having MSC2 molecule and present these antigens to the TH cell. And after the getting interaction with the uh, antigen with the binding of T cell with the help of T cell receptors, and the communication between these two cells, okay, either dendritic cell or macrophages, and communication between two receptors activates the TH cell, and activation of TH cell leads to the synthesis or production or degranulation of these, the release of the interferon gamma is the most important cytokinin, and which is having the uh, which has direct antiviral activity. Okay, interferon gamma having the direct antiviral activity against different types of cells. Also, and different types of infected cells, body cells, not other than those are not professional antigen presenting cells, but also those cells are also having the MSC molecule, that is MSC molecule 1, and this MSC molecule infected cells present the antigen with the help of MSC molecule 1, and all the nucleated cells are having MSC molecule 1. Whenever they got infected with the viral particle, they present the antigen with the help of MSC 1. Wherever the MSC1 is there with the with connecting with the antigens, and T cytotoxic cells come and contact with those cells. Okay? And what happens after the connection with the binding with the T cell with the anti viral infected cell? The T cell activation means TC cell activation will be taking place after binding with the infected cell in the acquired, especially cell mediated branch of immunity. And activation of TC cell may also leads to the release of the interferon gamma. Okay, interferon gamma, and this uh, besides the releasing of the interferon gamma, it also releases the different types of enzymes. For example, porphyrin granzymes are released from the cytotoxic cell and directly having the killing property of different infected cells, especially viral infected cells. And this is the important role of the Th cell and Tc cells in the killing of viral infected cells. Okay, we have another most important third cell, which is also playing a key role in the killing of the viral infected cells. That is natural killing cell and macrophages also there. Okay, both cells. Four types of cells are there in the cell mediated branch. Okay, which are playing a key role in the infect killing the or elimination of infected cells, especially viral infected cells. What is the natural kill? What natural killer cells do? Killer cells kill the virus of infected cells by antibody dependent cell mediated cytotoxicity. Okay, okay. This cells comes under may comes under cell mediated immunity, but also with the help of humoral immunity, especially with the help of antibody. Antibodies are guide the target, and these natural killer cells, macrophages, eosinophils, basophils, neutrophils, okay, are different types of cells uh, which are having the receptors on its surfaces for the uh, FC region of the different types of immunoglobulins. Whenever this FC region uh, binding with the FC region triggers the activation of the different types of cells what we have talking about natural killing cells, macrophages, eosinophils, basophils, neutrophils, like that. Different, especially macrophages also. What they do, they whenever the binding of the FC region to the FC receptor, FC region of the antibody to the FC receptor are on the surface of these cells, the activation may be triggers. Okay, activation will be triggering, and triggering of the different cell activation may lead to the different types of cytokines production, and different types of enzymes. Okay, different types of protein synthesis and enzyme releasing from the cells. Different types of mechanisms are taking place, which are very uh, dangerous for the infected cells. Okay, which are having the killing property to the infected cells. Okay, against the infected cells. So this these are the immunities against the direct viral infected cells. Okay, in the branch of acquired immunity, humoral immunity also play a key role along with the cell mediated immunity. So the dendritic cells, okay, dendritic cells play a key role in the activation of the different types of cells. With the whenever these dendritic cells get infected or get engulfed, having the property to the engulfing property, and after the infection, after the viral infection, these cells are engulfed by the dendritic cells by the process of phagocytosis. After processing of the antigens, these dendritic cells present the antigens to the Th cells. Okay, and Th cell, you know the story of what happens after the attachment to the dendritic cell MSC molecule to the Th receptor, T cell receptor, especially CD4. Okay, CD4 molecule. You know the remaining story. Okay, so Uh, this is about the viral uh, infection, immune response to the viral infection. Wait for 1 minute. I'm going to share one another image for your better understanding. So, I mean, saying error. So, after getting phagocytosis of the viral particle, it presents the antigen with the help of MSC molecule 2, and this MSC molecule 2 uh, presents the antigen to the TH cell, and TH cell releases the interferon gamma, and the interferon gamma activates the cytotoxic cell, okay, cytotoxic T cell, okay. Again, this cytotoxic T cell releases the interferon gamma, and this interferon gamma go and bind with the uh, other healthy cell, okay, other healthy cells other than infected cells, okay, healthy cells of host body, okay. Wherever the infection cells are there, those cells are killed by the uh, TC cell, and remaining cells, okay, healthy cells are having these interferon gamma receptor. So interferon gamma go and bind with the other healthy cells, and then these cells are not uh, getting any infection with the virus. That is the beauty of interferon gamma. After attachment with the interferon gamma with the surface receptors of the healthy cells, and these healthy cells might not get viral infection after binding with the interferon gamma. Okay, which cells releases interferon gamma? TH cell and TC cells are having the ability to release the interferon gamma. Okay, that is the main role of interferon gamma. Interferon gamma, whenever this interferon gamma attaches on the surface of receptors to the of the LD cell, and this LD cell uh, surface molecules arrangement will be changed, and changes may not allow the attachment of the plasma membrane, may not allow the attachment of the viral particle to the plasma membrane. That is the beauty of interferon gamma. So, and the remaining infected cells will be killed by the this uh, cytotoxic T cell, and different types of cells comes under the antibody dependent cell mediated cytotoxicity. This is how the viral this is how our immune cells or immune system respond against the or over the virus molecule. Okay, if you have any doubts you can ask now. Okay, immune response against virus molecules. If you have any doubts you can ask. No doubts, Amma?
No, sir.
Okay, so just read about the acquired immunodeficiency syndrome immune response against the AIDS. So I don't want to go into the details of the immune response of viral, especially HIV. I am going to give a brief account on infection of against the HIV virus. Okay? Immune response against the HIV virus. So exactly where exactly these HIV virus they're acting upon, exactly the significant depletion of CD4 T cells, resulting in the increasing susceptibility to different types of infections. And whenever this viral HIV virus comes and encounter on the different types of body cells, and always this HIV virus encounter on the CD4 T cells, okay, especially this CD4 T cells are having the affinity, okay, affinity to binding with the HIV molecule or HIV virus, the binding affinity, okay, binding affinity of the T cell will attract the HIV virus towards the T cell, okay, T cell, especially CD4 containing T helper cells. And the preference of CD4 cells is due to the high affinity they have mentioned in your book, page number 149. Due to the high affinity interaction between the a coat protein of HIV-1 and cell surface CD4 molecule, this high affinity may leads to the attraction or binding of the CD4 T cells with the with HIV molecule, HIV virus. So uh, we have different types of immune cells and different types of cells, but this especially HIV virus first of all prefers to bind with the T helper cells because of the high affinity surface molecules. Okay, these TH cells are having high affinity surface molecules on its surface. Because of this reason, the TH cell, HIV molecule, HIV virus infect the TH cell. Okay, TH cell, and beside the CD4 molecule and T cell co-receptors also helps to attachment THCR4 and different types of cells surface receptors also playing a key role in attachment and attracting the HIV virus towards the TC cell and TH cell, sorry, TH cell. And beside the TH cell and second target cell also be there for HIV virus. What are they? They are macrophages. Okay, they are macrophages because they are also having little bit affinity more than the other cells, unlike the TH cell, but it has little bit more than other remaining cells. Okay, except TH cell. What are the receptors involving in the attraction process? CCR5 receptor on the surface of macrophage. Okay, macrophages are having CCR5 receptor functions to attract the HIV virus towards the macrophages. Okay, and these receptors, affinity of these receptors may leads to the infection with the HIV molecule, okay, HIV virus. So you know the remaining story, how the diagnosis, how the HIV can be diagnosed, and the different types of stages and therapies. Okay, you know all those things and vaccination of current strategy. You know, just go through your book and you have hepatitis virus. Okay, just to go through these hepatitis virus. You have all these things in your previous UG and intermediate classes. So don't bother about the virus name. Just remember, just know the mechanism. Okay, just write down the mechanism. All the all the any if you study the mechanism of the response of immune system against the virus particle or virus molecules, you can understand. You can write everything. If it is HIV or hepatitis or other viruses, all the mechanisms are same, more or less. Okay, so don't bother about different names of viruses. Just remember the mechanism. Okay, what type of the response is working against the viral infection. Okay? We have innate branch of immunity, especially barriers. Okay, different types of barriers and different cells. Okay, natural killer cells and different types of cells also there, and T cells, TC cells, and different types of cells and antibodies also there. So you have you you can understand everything after understanding the innate immunity and humoral immunity and cell mediated immunity. And everything is possible after better understanding of these three topics. You can study everything. You can study the infectious diseases response. You can study cancer immunology, everything. You can study everything of immune system. So coming to the bacterial immune system response against bacteria. In this picture, what they have given? They given uh, bacteriolysis. Okay, bacteriolysis is the one of the mechanism to kill the bacteria. I will explain what exactly bacteriolysis after the brief account on giving the brief account on this image. And opsonization is one of the way to kill the bacteria to by the enhancing the phagocytosis. And one another mechanism is that antibody dependent cell mediated cytotoxicity. Okay. In this immune response, antibodies are play a major role to attract the or to kill the different types of the pathogens, especially bacteria. And agglutination is one of another mechanism to kill the uh, the different types of the bacterial uh, organisms. Okay, bacteria pathogens. And so neutralization of the toxins. Okay, and these are the products. Toxins are the products of bacteria, especially. And these toxins, okay, they may be exotoxins and endotoxins. These toxins are neutralized. Array, the neutralized with the help of different types of antibodies. After the neutralization, the antigen antibody complexes are formed, and the elimination of the uh, elimination mechanism for the antibody complexes. Okay, so we have another mechanism, secretion blockades. Okay, secretion blockades means with the help of different types of antibody. Don't bother about the secretion blockade. So we have different types of mechanisms. See the page number 156. The host immune response. The table has given one table has given 11 number table number 11.4. Host immune response to bacterial infection and bacterial evasion mechanism. An infection process is that attachment to host cell. Okay, what happens after attachment to the host cell? Host defense is that blockage of attachment by secretion of by secretory immunoglobulin A antibodies. So wherever the antibodies are pathogens are going getting into the uh, inner surfaces, the layer, the mucus layer is there, and this is the one of the barrier comes under innate branch of immunity, and this mucus layer is also having different types of immunoglobulin, especially immunoglobulin A. And this immunoglobulin A prevents the entry by blocking the antigen sites by attaching with the epitopes of the on the surface of the antigen. Neutralization will be will be seen after the attachment with the antigen epitopes of the paratope region with the help of with on which there in the antibody and secretion of proteases that cleaves secretory immunoglobulin A dimer. Okay? And evasion means escape mechanism. What is the escape mechanism? Secretion of proteases. Okay, our body respond by releasing the immunoglobulin A. And what bacteria do? Bacteria can also having the escaping mechanisms. For example, by secreting the proteases, okay, that cleaves the secretory immunoglobulin A dimer. That cleaves the immunoglobulin A dimer. Thereby, after the cleaving of the immunoglobulin A, bacteria enters into the system, okay, epithelial layer. Okay, after the destruction or the denaturing of the antibody A with the help of proteases, secreted by the bacteria, and this after the degradation of the immunoglobulin A, it into the get enter into the epithelial layer. So this is overall infection and host defense and bacterial evasion mechanism. And next one is that proliferation. Okay, and after entering into the body, it's try to proliferate into our cell. Okay, our system. Whenever the and pathogen, that is bacteria, enters into the cell tissues, the tissue area, our especially different types of macrophages, okay, are there in the different types of tissues. You know about the different types of macrophages, and these macrophages recognize the uh, entered bacteria and engulfs them. Okay, all the macrophages engulfs the bacteria, and after the engulfing, and it process the antigen areas on the surface of the bacteria, and it presents with the help of the MSC molecule too. Okay, and this is post defense. What bacteria do? The production of surface structures that inhibit the phagocytosis. Surface molecules can be changed. It changes its surface molecules. It inhibits the attachment of the phagocytic cells to the bacteria. Okay, bacteria releases or changes its surface uh, molecule arrangements. Okay, by this rearrangement or the production of different types of surface structures changes may leads to the attachment. Okay, and or inhibit the attachment of the macrophage or dendritic cells to the bacteria. Thereby, it inhibits the phagocytosis, and by this process, they escape from the phagocytic cells. Okay, this is the bacterial response, and phagocytosis is the host response. Okay? And after some time, what after the escaping from the phagocytic cells and complement protein, different types of complement mediated lysis will be there, and if it is escaped from the macrophages, okay, and different types of antibodies are synthesized or produced against the bacterial surface antigen, and these antigens goes and binds with the surface molecules of the bacteria, and whenever these antibodies are there are attached with the antigen antibody complexes are formed, and whenever these antigen antibodies complexes are formed, the complement proteins, okay, subunits get attached with the FC region, and this may leads to the complement activation. Okay. Especially complement proteins 3B, okay, subunit 3B, play a key role in the opsonization and different types of complement protein, complement pathways activation. So this is the host response. What is the host response? Complement protein attachment, okay, antibody attachment to the bacterial cell surface, and activation of complement protein attachment with the FC region, and activation of complement protein is the host response. Okay, this is the host defense or host response against the bacteria. And what bacteria do? Generalized resistance of gram-positive bacteria to complement-mediated lysis. Okay, and these surface molecules, okay, different types of surface molecules are of the gram-positive bacteria are might not be affected with the might not be recognized with the complement proteins. So it also has a escaping mechanism. Escaping mechanisms are developed in the bacterial cells to uh, our host immune response. Okay. different types of bacteria are developed different types of escaping mechanisms. Excuse me. So, insertion of membrane attack complex prevented by long side chain in cell wall, some gram negative bacteria, different types of whether the component activation is taking place at the time of uh, membrane attack complex formation with the help of different types of mechanisms which are present in the bacterial cell. Okay. Bacterial cells are having the different types of capacities or different bacterial cells develop the different types of mechanisms, escaping mechanisms uh, for the host immune response. And next, invasion of host tissue. Okay, if bacteria escaped from the complement system and the phagocytic system and phagocytic mechanism, complement mechanism, if it is escaped from the immune globulin A, and after this barrier, our antibody mediated agglutination. Okay, different types of antibodies are there, are released, get attached with the on the surface of the uh, antigen, especially different types of. Sorry. So, antibody mediated agglutination will be taking place, especially immunoglobulin G, immunoglobulin M play a key role in the agglutination process. And this process also can be inhibited or escaped by the bacteria by developing the secretion of elastases. Elastases that inactivates the 3CA and C3A molecule of a subunit of the complement protein and C5A molecule of complement protein 5. So these two molecules play a key role in the agglutination process of the bacteria, and the bacteria develop one beautiful mechanism. What is that? Secretion of elastases. Okay, and different types of elastases secreted towards the against the C3A molecule and C5A molecule, and these molecules can be inhibited after the attachment encounter by the elastases, and the agglutination of the different types of bacteria can be stopped. Okay, agglutination mechanism can be stopped. And how our body responding against the bacteria, and how the different types of the mechanisms are developed, how the bacteria escape from these immune responses. That is the very important. And so we have another step, that is toxin induces damage to host cell. Okay, toxin induced damage to host cell after the escaping from above mentioned mechanisms. Different types of other mechanism also develop. Neutralization of toxin. Okay. After the escaping from the above mentioned mechanisms, they undergoes the proliferation and start releasing of the different types of toxins. So those toxins can be neutralized by the different types of antibody release. Okay, these antibodies goes and binds with the toxin and neutralizes all those toxins. That is one and only way to prevent the uh, disease causative symptoms. Okay, this is innate immune branch uh, mechanisms are involving in the elimination or killing of the bacteria, same like viruses, and also humoral immunity branch also can be involved in the elimination of the killing of the bacteria along with the cell mediated immune branch. This is about bacterial immune response against bacteria. Okay, simple thing. Okay, in every process, in every immune response, you you can recall. We will also study about the uh, you may have
హలో
innate immunity. Excuse me.
హలో సార్, ఎక్స్క్యూజ్ మీ
Uh,
సార్. సార్,
tell me.
ఒక్క 2 నిమిషాలలో అంటే మేము చిన్నగా బ్రీఫ్ గా ఒక్కసారి
Um,
హ్యూమోరల్ అండ్ సెల్ మీడియేటెడ్ ఇమ్యూనిటీ గురించి ఒకసారి చెప్తారా సార్? అది ఎప్పుడు కన్ఫ్యూజ్ అవుతుంది నాకు.
humoral and cell mediated immunity. Amma?
సార్, అవును సార్.
Okay. 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 So, because of the importance of humoral and cell mediated immunity, you must know about this difference of these two immunity also, immunity branches. In the humoral immunity, antibodies, antibodies are play a key role. Okay, in the humoral immunity. Okay, especially B cell play a key role. And, most of the times, B cell play a key role in the humoral immunity uh, by releasing of antibodies. Okay, antibody ఉంటుంది ఇంకా హ్యూమరల్ ఇమ్యూనిటీ అంటే యాంటీబాడీస్ హెల్ప్ తోని జరిగేది విత్ ద హెల్ప్ ఆఫ్ యాంటీబాడీ ఓన్లీ. ఓకే, అండ్ కమింగ్ టు ద సెల్ మీడియేట్ ఇమ్యూనిటీ, cells are involving cells are involving in this type of cell mediated immunity, especially cells means T cells, okay? Th cell and Tc cells. Sometimes in this interlink between two branches also be there. Both both immunity by with the help of both immunity branches, the different types of pathogens and parasites will be killed. But the main difference is that the B cell comes under humoral immunity, especially antibodies. All the all the humoral immunity branch talks about antibody importance. Okay, antibody importance towards the elimination of the different pathogens. And what cell-mediated immunity talks? Cell-mediated immunity talks about the TC cell, the importance of the TC cell and TH cells. Okay, and especially TC cell, a TC cell and TH cell. What a TC cell do? TC cell, whenever the TC cell recognizes the antigen on the surface of the nucleated cell. Okay, nucleated means all the cells, and it cannot bind with the professional antigen cells, antigen presenting cells like macrophages and dendritic cells, B cells. It cannot having the property property to attach with these cells, but it can only bind with the other than professional antigen presenting cell. Each and every nucleated cells are having MSC molecule 1. Those cells for that cell, our immune cell, that the T lymphocyte, cytotoxic T cell only binds with that cells. Okay. Remaining T cell cannot do that process. Remaining T cell, TH cell, remaining means TH cell cannot having that property. Okay, because TH cell only binds with the TMHC2 molecule cells. Okay, which are not there in all every surface cell, every cell of the body. Only and
సార్,
professional
ఒక మాట
antigen.
సార్.
చెప్పండి.
సార్, సార్ ఇప్పుడు
చెప్పండి.
హ్యూమోరల్లో అంటే ఒకవేళ యాంటీజెన్ మన బాడీలోకి వెళ్ళినప్పుడు మన ఫస్ట్ లైన్ సెకండ్ లైన్ ఫెయిల్ అయినప్పుడు థర్డ్ లైన్ మెయిన్ ఇమ్యూనిటీ స్టార్ట్ అయినప్పుడు ఏమవుతుంది? ఆ యాంటీజెన్ ని పట్టుకోవడానికి హ్యూమోరల్ అంటే బి సెల్స్ అనేవి ఫైట్ చేస్తాయి. ఈ బి సెల్స్ ని సపోర్ట్ చేయడానికి టీ సెల్స్ వస్తాయి.
సపోర్ట్ ఏం లేదు, ఇంకా బి సెల్స్ కు సపోర్ట్ చేయడానికి టీ సెల్ వచ్చిందంటే అది సెల్ మీడియేటెడ్ అయిపోయింది. బి సెల్ స్పెషాలిటీ ఏంటంటే బి సెల్ స్పెషాలిటీ ఏంటంటే డైరెక్ట్ గా యాంటీజెన్ ని గుర్తిస్తే అది హ్యూమరల్ బ్రాంచ్ అవుతుంది. దట్ ఈస్ మెయిన్ డిఫరెన్స్. నేను అదే వస్తుండే నెక్స్ట్ స్టెప్ లో. బి సెల్ డైరెక్ట్ గా యాంటీజెన్ ని గుర్తిస్తే అది హ్యూమరల్ రెస్పాన్స్ కిందకి వస్తుంది.
ఓకే సార్.
ఇది డైరెక్ట్ గా యాంటీజెన్ తో కమ్యూనికేట్ అయ్యి యాక్టివేట్ అయితే అది హ్యూమరల్ బ్రాంచ్.
ఓకే సార్.
సమ్ టైమ్స్
ఒకవేళ
టీహెచ్ సెల్
ఇప్పుడు
ఆల్సో టీహెచ్ సెల్ తోని
THL కూడా
బైండ్ అయ్యి యాక్టివేట్ అవుతే అది యాంటీబాడీస్ రిలీజ్ అవుతాయి. కానీ అది మనం హ్యూమరల్ బ్రాంచ్ కింద రెండు ఇంటర్లింక్ అనమాట రెండిటికి.
ఓకే
మెయిన్
సార్.
మొత్తం మనకు డైరెక్ట్ గా యాంటీజెన్ ని గుర్తిస్తే అది కంప్లీట్ గా హ్యూమరల్ బ్రాంచ్ వస్తుంది.
ఓకే.
టీహెచ్ సెల్ ఎల్పిఎస్ ఎప్పుడంటే అది మన మ్యాక్రోపేజ్ తో కానీ డెంటటిక్ సెల్ తో కానీ బైండ్ అయినప్పుడు మాత్రమే ఇక్కడ బి సెల్ అవసరం ఉంటుంది. మోస్ట్ ప్రాబ్లీ అది టీసీ సెల్ ని యాక్టివేట్ చేస్తుంది టీహెచ్ సెల్.
ఓకే.
అర్థమైందమ్మా? ఒకవేళ డైరెక్ట్ గా
ఓకే సార్.
యాంటీజెన్ ని బి సెల్ గుర్తించలేకపోతేనే ఈ టీహెచ్ సెల్ అనేది హెల్ప్ చేస్తుంది. అంతేగాని డైరెక్ట్ గా గుర్తించే యాంటీజెన్స్ తోని ఇది హెల్ప్ చేయదు.
ఓకే. ఒక యాంటీజెన్ ని
మో-
డైరెక్ట్ గా బాడీ గుర్తిస్తే అప్పుడు బి సెల్ ఏం చేస్తుంది?
ఆ
బి సెల్ దాన్ని చంపేస్తుంది. ఇన్ కేస్
బి
కొన్ని
సెల్
కేసెస్ లో ఇప్పుడు మనకి కొత్త వైరస్లు ఏమైనా వస్తే అది ఒకవేళ బి సెల్ అంత ఫాస్ట్ గా ఐడెంటిఫై చేయకపోతే టీహెచ్ సెల్ హెల్ప్
అది
చేసి బి సెల్ ని యాక్టివేట్
అదే
చేస్తుంది.
ఇంకోటి అంటే
ఓకే
ఫస్ట్
సార్.
సెల్ మీడియేటెడ్ ఇమ్యూనిటీ ఫస్ట్ రెస్పాండ్ అవుతుందమ్మా. ఎందుకంటే డిఫరెంట్ టైప్స్ ఆఫ్
ఓకే సార్.
యాంటీజెన్ ప్యారెంటిక్ సెల్స్ ఉంటాయి. నెక్స్ట్ రెండో
ఎస్ సార్.
ఎన్కౌంటర్ లో హ్యూమరల్ ఇమ్యూనిటీ ఎగ్జాక్ట్ గా పనిచేస్తుంది.
ఓకే. ఒకసారి అది ఐడెంటిఫై అయిపోయిన తర్వాత ఇంకా బి సెల్ అనేది రంగంలోకి దిగి ఫుల్ గా చంపేస్తుంది వాటిని.
మెమరీ సెల్స్ ఉంటాయి కాబట్టి ఇంకా కొన్ని
ఓకే.
సార్లు అది వే ఆఫ్ సర్ఫేస్ మాలిక్యూల్ వే ఆఫ్ యాంటీజెన్ యాంటీజెన్ ప్రాపర్టీ అనమాట. ఇక డిఫరెంట్ టైప్స్ ఆఫ్ ప్రాపర్టీస్ ఉన్నాయి. అది టీహెచ్ సెల్
ఓకే సార్.
యాంటీజెన్ ప్రెజెంటింగ్ సెల్స్ ఉన్నాయి. యాంటీజెన్
ఉమ్.
ప్రెజెంటింగ్ సెల్స్ గుర్తిస్తే టీహెచ్ సెల్ మీడియేటెడ్ ఇమ్యూనిటీ అయితే,
ఓకే
బి
సార్.
సెల్ గుర్తిస్తే అది హ్యూమరల్ అయితే. ఎక్కడికైనా
హ్యూమరల్ అవుతుంది.
యాంటీబాడీస్ మేజర్ రోల్ తీసుకుంటే అది హ్యూమరల్ బ్రాంచ్.
ఓకే సార్.
టీసీ సెల్ టీహెచ్ సెల్ ఉంటే ఎస్పెషల్లీ టీసీ సెల్ అండ్ మ్యాక్రోపేజ్ సెల్. ఓకే, టీహెచ్ సెల్ ఆల్సో యాక్టివేట్ ది మ్యాక్రోపేజ్. టీహెచ్ సెల్ బి సెల్ ని యాక్టివేట్ చేస్తుంది, టీసీ సెల్ ని యాక్టివేట్ చేస్తుంది, మ్యాక్రోపేజ్ ని కూడా యాక్టివేట్ చేస్తుంది. సో ఇక్కడ బి సెల్ ని యాక్టివేట్ చేయకుండా టీసీ సెల్ ని మ్యాక్రోపేజ్ ని కంప్లీట్ గా రెండు సెల్ ని యాక్టివేట్ చేస్తే కంప్లీట్ గా సెల్ మీడియేటెడ్ ఇమ్యూనిటీ అవుతుంది. ఈ బి సెల్ అనేది ఇది ఇంటర్లింక్ అనమాట. బి సెల్ అంటే
ఓకే సార్.
హ్యూమరల్ అది కంప్లీట్ గా యాంటీబాడీస్ తోనే అది కంప్లీట్ గా క్యూర్ అవుతుంది అంటే అది హ్యూమరల్ బ్రాంచ్. అది అట్లా పాసిబుల్ కాదు. ఈ సెల్
ఓకే
టీసీ
సార్.
సెల్ హెల్ప్ కావాలి, మ్యాక్రోపేజ్ హెల్ప్ కావాలి, అలాంగ్ విత్ బోత్ ఇమ్యూనిటీ ఫైట్ అగేన్స్ట్ ది డిఫరెంట్ టైప్స్ ఆఫ్ ఇన్ఫెక్షన్. ఆ రెండు యూనిట్ లో యాంటీబాడీస్ తోని మనం ఫైట్ చేస్తే అది మనం హ్యూమరల్ అంటాం. టీసీ సెల్ మ్యాక్రోపేజెస్ వస్తే అది సెల్ మీడియేటెడ్ అంటాం.
ఓకే సార్.
మనం వేరు చేయలేము ఈ రెండింటిని, కాకపోతే మెయిన్ డిఫరెన్స్ అది.
ఓకే సార్. రైట్ సార్. థ్యాంక్
ఓన్లీ
యూ సార్.
యాంటీబాడీస్ వస్తే ఓకే అమ్మా. సో
ఓన్లీ యాంటీబాడీస్ వస్తే
ఓన్లీ యాంటీబాడీసే రావాలి, అది హ్యూమరల్ ఇమ్యూనిటీ.
ఓకే సార్.
ఇక్కడ సైటోటాక్సిక్ సెల్ మ్యాక్రోపేజెస్ వస్తే అది మనం సెల్ మీడియేటెడ్ ఇమ్యూనిటీ బ్రాంచ్ మాట్లాడుతాం. ఫర్ ఎగ్జాంపుల్ ఇక్కడ మీకు వైరస్ గురించి ఎక్స్ప్లెయిన్ చేసినప్పుడు పేజ్ నెంబర్ 147 అన్నాడు.
ఎస్ సార్.
147 లో రిమైనింగ్ టేబుల్లో వాళ్ళు ఏమి ఇచ్చారు? ఓన్లీ టీహెచ్ సెల్, టీసీ సెల్, మ్యాక్రోపేజ్, న్యాచురల్ కిల్లర్ సెల్ ఇచ్చారు. ఈ సెల్స్ కిల్ చేస్తే అవి సెల్ మీడియేటెడ్ అంటాం అంతే. అది ఒక్కోసారి యాంటీబాడీస్ వచ్చి అటాచ్ అయితేనే కొన్ని సెల్స్ కిల్ చేస్తాయి. డైరెక్ట్ టీసీ సెల్ రోల్ తీసుకుందంటే అది సెల్ మీడియేటెడ్ ఇమ్యూనిటీ, కంప్లీట్ సెల్ మీడియేటెడ్ ఇమ్యూనిటీ అవుతుంది.
ఓకే.
కన్ఫ్యూజ్ అయ్యారమ్మా మళ్ళా?
లేదు సార్, లేదు సార్.
ఏం లేదు, జస్ట్ మీరు యాంటీబాడీస్ హ్యూమరల్ ఇమ్యూనిటీ అంటే యాంటీబాడీస్ రిలీజ్ అయ్యి అవి న్యూట్రలైజ్ చేస్తాయి. అవి డిఫరెంట్ టైప్స్ ఆఫ్ యాంటీబాడీస్ ఫంక్షన్ అన్ని కూడా హ్యూమరల్ ఇమ్యూనిటీ కిందకి వస్తాయి. అలా కాకుండా టీఎస్ సెల్, టీసీ సెల్ లాస్ట్ స్టెప్ లో బి సెల్ ని యాక్టివేట్ చేస్తుంది. అక్కడ మీకు కన్ఫ్యూజన్ ఉండొచ్చు, కానీ మీరు అక్కడ వరకు వెళ్ళకుండా టీసీ సెల్ ని యాక్టివేట్ చేస్తుంది, మ్యాక్రోపేజ్ ని యాక్టివేట్ చేస్తుంది. అట్లా మీరు ఐడియా ఉంచుకోండి. టీసీ సెల్ యాక్టివేట్ అయినా సెల్ మీడియేటెడ్ ఇమ్యూనిటీ అవుతుంది, మ్యాక్రోపేజ్ యాక్టివేట్ అయినా సెల్ మీడియేటెడ్ ఇమ్యూనిటీ అవుతుంది.
సెల్ మీడియేటెడ్ అవుతుంది. ఓకే సార్.
రెండు కంప్లీట్ గా సెల్ మీడియేటెడ్ ఇమ్యూనిటీ కిందకి వస్తాయి.
ఓకే
సో
సార్.
డిఫరెంట్ టైప్స్ ఆఫ్ మెకానిజమ్స్ అనేవి అన్ని కూడా ఈ 3 బ్రాంచ్ల కిందకే వస్తాయి. ఇన్ఫ్లమేషన్ అనేది ఇన్నేట్ ఇమ్యూనిటీలో ఉంది. ఓకే, ఇన్ఫ్లమేషన్ ఇస్ వన్
ఓకే
ఆఫ్ ది
సార్.
మెయిన్ ప్రాసెస్ in under the innate immunity branch. Phagocytosis is a major mechanism under the innate immunity branch. And also classical pathway, okay, complement system example. Complement system అన్న ఎన్ని మన ఇన్ఫ్లమేషన్ అన్న ఈ యొక్క ఇంకేం చెప్పినాం మనం, phagocytosis along with the barriers, phagocytosis, inflammation, and the complement system comes under innate branch of immunity. Okay, na? Though they are coming under
ఓకే సార్.
innate immunity branch, for the activation of the system, okay, for the activation of the system, it requires the antibody attachment to the on the surface of receptor. ఇవి innate branch లకు వచ్చినప్పటికి కూడా యాంటీబాడీ అనేది అటాచ్ కావాలి కొన్నిసార్లు. కాంప్లిమెంట్ యాక్టివేషన్ అంటే యాంటీబాడీస్ అవసరం ఉంటే ఒక మెకానిజం, క్లాసికల్ మెకానిజం. అట్లానే మనం దీన్ని మళ్ళీ తీసుకెళ్లి అక్వైర్డ్ ఇమ్యూనిటీలో వేయలేం, ఇమోరల్లో. సమ్ టైమ్స్ దే వర్క్ along with other, they work combinedly, but completely barriers, complement system, and inflammation, phagocytosis comes under innate branch. And in the humoral branch, different types of antibodies, action of different types of antibodies. For example, immunoglobulin A, E, G, M, different types of immunoglobulins comes under this humoral branch. And cells, macrophages, natural killer cells, and TC cell comes under TH cell comes under cell mediated branch. Okay, for the activation they requires different types of molecules. Though they require different types of molecules, they comes under different types of branch. యాంటీబాడీ వచ్చింది కదా యాక్టివేషన్ కు అంటే అది హ్యూమరల్ కాదు. ఓకే, హెల్ప్ చేయడానికి యాంటీబాడీ వెళ్తుంది అంతే. అలా అని మనం హ్యూమరల్ వేయలేం. యాంటీబాడీస్ రిలీజ్
ఓకే
అయిందంటే
సార్.
డిఫరెంట్ టైప్స్ ఆఫ్ మెకానిజమ్స్ ఉన్నాయి దానికి. ఒక యాంటీబాడీ రిలీజ్ అయిందంటే అది డిఫరెంట్ వేల దాని ఇన్ఫెక్షన్ తగ్గించడానికి ట్రై చేస్తుంది, న్యూట్రలైజేషన్ చేస్తుంది. ఓకే, ఆప్షనైజేషన్ చేస్తుంది, కాంప్లిమెంట్ ని యాక్టివేట్ చేస్తుంది. అంటే యాంటీబాడీ కాంప్లిమెంట్ యాక్టివేట్ చేస్తుంది అంటే కాంప్లిమెంట్ సిస్టమ్ తీసుకెళ్ళి మనం యాక్టివేట్ ఇమ్యూనిటీలో వేయలేం. కనెక్షన్ అండి, ఇంటర్లింక్ బిట్వీన్ all it has interlink between all the immunity branches. అవన్నీ డిఫరెంట్ అయినప్పటికీ ఇంటర్లింక్ ఉన్నాను. ఇందులో మాలిక్యూల్స్ దానికి అవసరం, దాని మాలిక్యూల్స్ దీనికి అవసరం. మనం క్లియర్ గా ఏం గుర్తుపెట్టుకోవాలంటే హ్యూమరల్ అంటే యాంటీబాడీస్ వస్తాయి, మొత్తం యాంటీబాడీస్ వస్తాయి, మెకానిజమ్స్ అవసరం లేదు. అలా యాంటీబాడీస్ రిలీజ్ అయిన వరకు హ్యూమరల్ ఇమ్యూనిటీలో వస్తాయి. సెల్స్, టీసీ సెల్, టీహెచ్ సెల్, మ్యాక్రోపేజ్, న్యాచురల్ కిల్లర్ సెల్, సెల్ మీడియేటర్లో వస్తుంది. phagocytosis, complement, inflammation, different types of barriers అన్ని కూడా innate లో వస్తాయి. క్లియర్?
ఎస్ సార్.
ఇంకేమైనా డౌట్స్ ఉంటే అడగండి అమ్మా.
Sir, is
లేదు.
apoptosis is exhibited by bacterial invasion also?
Apoptosis also exhibited
By bacteria?
due to the bacterial infection by
Our
the
host
TC
body
cell.
initiates
Ah.
apoptosis, sir, but bacteria also
our
inducts apoptosis in macrophages?
bacteria cannot induce this apoptosis mechanism, but our TC cell and different types of enzymes induces apoptosis process by the activating the caspase 8 enzyme. Okay, inactive caspase 8 enzyme can be activated by the different types of molecules which are released by the macrophages and T cytotoxic cell. For example, granzyme is the enzyme which is released by the T cytotoxic cell. It enhances or stimulates the caspase 8 enzyme. By the stimulation of the caspase 8 enzyme, natural apoptosis of infected cell will be happen. Bacteria cannot directly cannot having the activation process, but the macrophages and enzymes of macrophages and cytotoxic cell having the direct connection with the apoptosis process.
Why? Because in page number 156, under proliferation, in the bacterial evasion mechanisms, induction of apoptosis in macrophages he has given. That's why I got the doubt, sir.
Where? 156 page.
156.
In the
under
table?
bacterial evasion mechanisms. Bacterial evasion
bacterial
mechanism under proliferation category, that induction of apoptosis in macrophages.
Induction of apoptosis in macrophages.
Yes, sir.
Induction of apoptosis in macrophages. Okay. Natural death of macrophages can be happen. Okay, by the releasing of the different types of defective invasion mechanism. It can be possible. They have given macrophage apoptosis can be
Induction by
happen.
bacterial evasion.
Yes, it can be happen. Our infected cell apoptosis by the cytotoxic cell, and macrophage apoptosis process by the bacterial products. Okay, it can be possible.
Okay, sir.
Very good question.
Thank you, sir.
Okay. Next.
Sir, for questions like immunity against bacteria, that means host immune system response against bacteria or virus, means if we write mechanism of this table and then structure, pathogenesis, diagnosis, immunity, treatment, prevention and control, is it okay, sir?
Yes, Amma, yes. Just give a mechanism. Give a mechanism. What is humoral mechanism against the humoral response of our immunity branch against the bacteria or virus? What are the what is cell-mediated immunity response? What is our innate immune response? Just give a brief account on these three branches, and write down the pathogenesis, diagnosis. Okay. If they ask for long question, do not to write everything for short question. If they ask under the long question, you can write everything.
Okay, sir.
Most of time focus on immune responses. Okay, more focus on immune response other than other areas like diagnosis, the immunity, pathogenesis. You need not write everything about the diagnosis and pathogenesis. Just focus on immune response. What is our immune response? Okay, what is host immune response against the different pathogens? That is important.
Okay, sir.
After the completion of the immune mechanisms, write down briefly about those things. Just majorly focus on the immune response of host against the pathogen. They expect more matter on about response only. More thing about response mechanisms. They expect more matter upon the immune response. So just to give a more information about the mechanisms, and write brief if you have a time, brief on diagnosis, okay, and pathogenesis, etc. Treatment. Okay, Amma?
Okay, sir. Thank you.
Yes. Any questions?
No, sir.
Okay. We will talk in immunity to parasitic diseases tomorrow class. In the tomorrow session, we will talk about immunity to parasitic diseases. Okay.
Last unit?
Okay.
Sir, what
Excuse
about
me,
last one?
Last one, immunodiagnosis. Okay.
Immunodiagnosis and applications.
If time permits, I will also complete those.
Overview, at least overview, sir.
I will give an overview about immunodiagnosis and application.
Okay,
Half
sir.
an hour, 40 minutes for half an hour to 40 minutes for parasitic diseases. And remaining time, I will give overview of immunodiagnosis, overview on immunodiagnosis and application.
Okay, sir. Thank you.
Okay, na? Okay, ma. If you have any doubts, you can ask tomorrow also.
Okay, sir.
All
Thank
the
you,
best.
sir.
I am going to learn. Okay, all the best.
Okay, sir. Thank you.